Hello, my name is Rafael Camillo. I'm a current first-year student in the joint doctoral program here at San Diego State, and currently I'm rotating within the house laboratory. And the laboratory studies ovarian cancer and the different signaling pathways responsible for the survival of tumors. So here we have a picture of a uterus, and here we have one of the ovaries. And within the ovary, in the case of cancer, we get these tumors. And within these tumors, these tumors are made of different types of cells. They're heterogeneous groups of cells. So we have different varieties pictured here. And when patients get diagnosed with ovarian cancer, they undergo chemotherapy, which is the treatment used to hopefully get rid of these tumors. And so with traditional chemotherapy up to this point with ovarian cancer, it knocks out most of the cells, but for some reason, some of them seem to survive the first round of chemotherapy. And so these cells survive, and eventually you get the relapse, where they're able to regenerate, and eventually the tumor comes back with all the different types of cells. And so these particular cells are of interest. We call these cells the tumor initiating cells. Or the TICs. And these tumor initiating cells have a number of different properties from standard cells. For instance, they're like most cancer, or any cancer, the rate of cell division is dysregulated, it's increased. And in this and also in ovarian cancer, we see these formations of what's called these spheroids. These spheroids. And typically these spheroids are a sign of metastasis. The problem with ovarian cancer is that it's typically detected within the stage 3 or 4 when the metastasis has occurred. So the goal is to somehow study the signaling pathways to hopefully detect it earlier before the stage 3 or 4 to increase the chance of survival within the patient. And these traits, one of the cytokines that causes these traits to occur, it's what's called tweak. And the cytokine named tweak, it causes these traits to occur by activating the alternative NF-kappa B pathway. And so previous experiments in the lab have grown these cells like in vitro. And so there are these 2D conditions, and we have these 3D conditions. And in the 3D condition, we can mimic the formation of these same spheroids. So they're mimicked here. But the problem is, previous experiments have noticed this upregulation of this receptor called FN14, which is also the tweak receptor. And so the question became, is that necessarily because we're just the media itself, as we're growing into 3D media, or one of the common components to this media is the fibroblast growth factor, or the FGF. So the question became, what is causing this? Is it just because of the media itself, or is it because of FGF? And so I set out to answer a more logistical problem about this by setting up an experiment by growing 2 separate cell lines, this OV90 SH-neg and the CaOV4 ovarian cancer cell lines. I grew them, extracted the whole cell lysate, and then performed a Western blot looking for this FN14 within, again. So here we have the 2D plus minus this FGF, and the 3D of the FGF, which is we have the minus plus minus plus. This is 2D, this is 3D, and this is one of the cell lines. And similarly, we have the same thing for the other cell line, the 2D and the 3D. And from my experiment, after running it multiple times, I found that there is no significant difference caused by the presence of this FGF within the media. So that can be applied to previous experiments that have been run using this 3D media. And so at this point, now that we've studied how we're growing these cells, now to get back to the bigger question at hand, how are we looking at tweak itself? So currently I'm beginning a project which looks at the interaction between tweak and the FN14, which is the tweak receptor. We're looking at how these interactions occur. One notable experiment I'm starting right now is the siRNA transfection, in which we are knocking down FN14 in the presence or in the absence of tweak, and similar in the 2D or the 3D media. And then from there, seeing how other, let's say, cytokines or other genes are affected when FN14 has been knocked down. So, once I'm able to grow I'm currently growing the cells right now. Once we grow these cells, then we're able to, once again, perform the whole cell lysate, but then do a fractionation to see the localization relative. Are these other related genes, such as, I believe that's called, this RelV, other factors, do they localize within the cytoplasm, toward the nucleus, and is that affected by this affecting the FN14 project? So, thank you. How do you?